Okay, I'll just have to shout. Okay, so hi everyone. Yeah, as I was kindly introduced, I'm going to be talking about the use of the biowave to optimize plant tissue embedding for volume EM. So before diving into the details, I'd just like to briefly introduce the main goal of the work. So volume EM is a powerful tool for studying intricate structures of cells. However, preparing plant tissues for volume EM presents its unique challenges. So our goal with this research was to improve how we embed plant samples using a microwave tissue processor called the biowave. And this significantly speeds up preparation time. But first, let's look at some of the challenges specific to plant tissues. I found this online and I thought it was very, very appropriate. So you must be the one for me since my selectively permeable membrane let you through. So working with plant tissues is quite different compared to animal tissues. Plant cells have complex cell structures like rigid cell walls, large vacuoles, and they vary greatly between different tissue types. This makes it difficult to prepare samples quickly and accurately, and compared to traditional preparation methods, it can take up to a week. So if something does go wrong, this can then be labor intensive to repeat the process. So this is where the biowave comes in, and it has the potential to address these challenges. So what exactly is the biowave? So the biowave is a microwave tissue processor that helps to speed up the embedding process by using programmable protocols, applying a vacuum-assisted infiltration, and this will help to infiltrate the reagents and then therefore reduce overall processing times. These features make it particularly useful for reducing sample time, which is especially important when dealing with large numbers of samples. So while we primarily use it for plant tissue embedding, it has many other applications such as immunolabeling, paraffin tissue processing, and decalcification. So let me just show you how we've been using the biowave in our study to try and optimize the embedding process. So this is a protocol that we initially tested, and it has been previously used and successful for plant samples in the biowave. So this was an obvious starting point since it has shown reliable results in plant tissue preparation for volume EM. So the key steps here will include the microwave-assisted fixation, dehydration, and resin infiltration. And the advantage of the biowave here is for its ability to accelerate these steps, potentially shortening the process that can normally take days into just hours. So what I've put here at the bottom, I've just highlighted some points of how quickly some of these steps will be. So the fixation steps could take no longer than 14 minutes, and for the further staining steps, 14 minutes, 7 minutes. So it's a very, very quick process compared to our manual protocols that we would use. And after trialing this out, these were the results. So as you can see, we've got some lovely contrast here. The protocol seemed to work beautifully, and I was very, very pleased with this. And for those that aren't familiar with plant science, so as you can see, these are just images of cells, these kind of large spaces in between are the large vacuoles that I was speaking about. We have the chloroplasts inside and the starch grains inside. So these are kind of the main things that we want to see. So this shows great fixation. The cells are bound to the cell wall, everything's contrasted lovely. So then recently I had someone, a friend of mine, Ned Deref from EMBO in Heidelberg, and he came and gave a talk discussing his serial block face imaging in mosquito gut. And this was the protocol that he used. And to my surprise, the contrast and staining worked beautifully. However, I noticed that in his protocol, there was no use of uranyl acetate or lead aspartate. So I thought, oh, this looks interesting. We've got the biowave. I'd be interested to see how this looks in the plant cells. So I decided to adopt this method for our plant tissue samples, as I was interested to see whether this could offer similar benefits for plant tissue imaging. And this was the result. So not as nice as the first round. As you can see, the fixation here is horrendous, but on the positive side, we have good contrast and staining. So I thought perhaps this might be a fixation problem rather than a issue with the contrast itself. And if I just quickly go back to the protocol, as you can see at the bottom, the steps were significantly longer to the traditional protocol that we use. So these were much, much longer, which made me think perhaps the fixation might have been a bit too aggressive for our plant samples as they are very delicate. So I thought, okay, well, we'll try and fix the traditional way and do an overnight fixation instead. So we did that and then continued the process in the BioWave using that same protocol. And these were the results. So as you can see, this is much more improved. The staining has worked beautifully. And this initial fixation step using the BioWave showed to be suboptimal, but manually fixing the samples overnight did produce better results. And what's exciting is that we could achieve good staining results without the use of uranyl acetate and lead aspartate, which are typically key heavy metals for staining. So this opens up possibilities for reducing hazardous material waste. So what does this mean for the broader field of plant EM? So overall, this work can contribute to making plant tissue preparation for volume EM a lot more efficient and environmentally friendly. Whilst the BioWave's fixation still needs improvement, the faster processing time and reduced reliance on heavy metals does show great promise. So this method could make volume EM studies more accessible for a wider range of plant samples, including rare plant species, which have been a challenge in the past. And this will then in turn lead to potentially more volume EM studies. So even though we've made progress, there are still some remaining challenges. So plant tissues are highly variable and no single protocol will work for every plant type. It's important to reduce artifacts whilst preserving the ultrastructure of these samples. And we also need to continue to refine these protocols for the BioWave testing to maybe test on a broader range of plant material to ensure consistent and high quality results. So to sum up, here are some of the key takeaways of the study. So the BioWave enabled official sample preparation, and it will continue to do so for future projects and for protocol development. So finally, I'd like to mention this piece of work that contributed to a recent publication in PNAS, where an Arabidopsis plant was engineered to develop a protopyrenoid to improve CO2 capture efficiency. So while the BioWave wasn't the leading primary focus of the paper, it did play a really important role by allowing us to prepare plant tissue samples more efficiently, which enabled us to achieve high quality results in a shorter time frame. So I'd like to take a moment to thank my collaborators from the John Innes Centre and the University of Edinburgh, as well as the bioimaging facility where the BioWave tissue processor played a really key role in the work. And I also want to acknowledge VBSRC for funding this project. And lastly, I just want to mention that we're currently hiring in the bioimaging platform at the John Innes Centre. So if you're interested in plant science research, feel free to scan the QR code or talk to me after if you'd like. So thank you so much for your attention, and I'd be happy to take any questions.
Thank you very much for this amazing talk. Do you have questions?
Thank you for the presentation. I wonder, have you explored the use of vacuum? Because in the BioWave, you can have different modes for, like, you know, and vacuum on, off, or
yes.
switch it.
Yeah, the vacuum, essentially we have it set on auto, which means it's kind of always on. I believe the vacuum doesn't come on for maybe the dehydration steps, but definitely for the fixation and for the staining steps, the vacuum is present.
And then are you, because if you do these osmication steps, then you see that there is a bit of osmium contamination that goes through the tubes inside of this machine. Mm-hmm. So what are you feeling on this?
Oh yes, yeah. Our tube is very, very black from the osmium. Yes. Yeah. So we in that sense, I don't know. I guess it's just something that will come of it. I know it feels a bit odd because when you have your samples in the BioWave, which we would traditionally, you know, kind of bottle. Kind of like put a cap on over our samples, so you know the osmium's kind of kept away. But when we're using the BioWave, we actually have them in Eppendorfs, and the Eppendorf lids are open, so that can feel a bit risky. I know I mentioned about obviously we omit the use of hazardous materials, but I haven't worked out a way to omit osmium. Yeah. But yeah, that is something that does happen, and I think I don't know if there's a way around that.
We still have time for questions.
Thank you. I have one. We are still recommending for plants the spore or viscosity spore. Can you comment this resin? Do you have some experience with it?
Not yet. So primarily in my role at the John Innes Centre, I was kind of brought in to start up boiling vEM, should we say, because it was a new technique that was brought into the platform. So I was told, this is what we're going to do, and here's Didcott. So I've been having the joys of working with Didcott. I've heard a lot of good things about Spurs resin, and we've actually got a Spurs kit, so that's something I need to work with. But I have heard that Spurs is, despite being also viscous, I don't know if Didcott's one of the more difficult ones to work with, but that's something I'll definitely explore, and we can have a talk afterwards for sure.
Any other question?
Not yet, but I would be open to that. I know for plant tissue material, sodium cactylate is kind of a key buffer to use. So that could be something to explore for sure, but we haven't looked into doing that just yet. But I would be open to any suggestions.
I was just wondering, have you had a go with some of the Reynolds type stains, so the lanthanide type things in place of?
Not me personally. I believe we do have one of those stains in our lab that we have used for post staining on TEM sections, and they seem to actually, it seems to work okay. I haven't actually trialed it yet for on block staining in the biowave, but that could be something to try for sure. But if I could kind of eradicate the use of it altogether, then why not?
Yeah, absolutely.
Yeah.
Thank you.
Thank you
for
very
your
much.
questions. Thank you.